All right, welcome back. So there are a few things here to mention about this overall system. The first is, in this particular interaction that we've just done, we've used acetylcholine as the neurotransmitter that communicates between the motor neuron and the effector cell, in this case, a muscle cell. Acetylcholine, very common neurotransmitter in this type of system, but we have to think a little bit about what's happening when the acetylcholine binds to a receptor. So if I take acetylcholine plus an acetylcholine receptor, I get acetylcholine bound to the receptor. The more acetylcholine I have, the more acetylcholine I should have bound to the receptor. That's because ultimately receptor ligand interactions overall are basically dynamic equilibria. The way you get more acetylcholine bound to the receptor is to increase the amount of acetylcholine that you have. Okay? This model also tells us that at any given time, given a certain number of receptors and a certain amount of acetylcholine, that there should be a certain proportion of this acetylcholine bound to receptor as well as some that isn't. But at any given point in time, even though this percentage should stay relatively constant, the individual acetylcholines are doing different things. Some free acetylcholine bumps into a receptor to form the acetylcholine receptor complex, and at the same time, some acetylcholine is falling off of receptors to produce more free acetylcholine. Does that make sense? There's always some acetylcholine becoming part of a receptor and always some falling off of the receptor, right? There's some forming and some coming off. That means that if the acetylcholine is coming off of receptors and more acetylcholine comes in, then if I'm an acetylcholine receptor here, let's give it a binding site for acetylcholine here, right? There's my acetylcholine receptor. This is the sodium channel that's activated by binding of acetylcholine. That means that if the acetylcholine binds, that flips open the channel and then the sodium should come in. If the acetylcholine falls off the receptor, which we've said that it can do, that would lead to closing of the receptor and no more sodium comes in. But another acetylcholine binds, flips open the receptor, and more sodium comes in. So I guess my point here is, whenever you have a receptor ligand interaction like this, it really isn't the ligand binds to the receptor and pops it open. It's the ligand binds to the receptor, pops it open, falls off, another one binds, flips open the receptor, falls off, another one binds. So you get this multiple bindings of multiple acetylcholines all happening within millionths of a second. So this receptor is being popped open. It's more like open, 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 open every time this binds. And as long as this acetylcholine stays in this synaptic cleft, I'm getting multiple activation of these sodium channels, which means the signal is continuing to be communicated. If the system were just acetylcholine binds to the receptor, it opens up the receptor, and we were done with it, then I would say that this signal has communicated with the receptor the following information: open up the receptor. On the other hand, what's really going on is that this signal is communicating multiple times per second with multiple receptors. It's not open up the receptor and you're done with it, it's open up the receptor, open up the receptor, open up the receptor, open up the receptor, open up the receptor. And as long as this acetylcholine stays in the synaptic cleft, that message continues to be delivered millions of times. And we don't want that. We just want the message delivered enough to trigger depolarization to ultimately pop open those calcium channels in the sarcoplasmic reticulum, and then we're done with it. We don't want to keep conveying this signal. If we're telling the muscle in the arm here to contract, that's what we want to tell it, contract. We don't want to tell it contract, 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 because then your arm's doing like this because it keeps contracting. Right? We want the signal to be delivered and be done with it. So in the acetylcholine system that's widely used, what we have is the ability to get rid of the signal. That's the motor plate. Here would be the neuron with the synaptic terminal. The acetylcholine is released from vesicles right into that neuromuscular junction. Along with this is an enzyme called acetylcholinesterase, which gets released at the same time. The acetylcholinesterase basically breaks up the acetylcholine into an acetate group, which is what this becomes, and a choline. Okay, you break acetylcholine into an acetate and a choline. The acetate derives from that acetyl group. The choline then gets recycled back into the neuron, and the neuron then rebuilds the choline back into acetylcholine by adding an acetate back to it. Okay? The acetate is not recycled back into the neuron. It ends up in the circulatory system, and that acetate can be metabolized for energy. Okay? So the idea here is once the acetylcholine is released, it binds to receptors, but the acetylcholinesterase is trying to take apart that signal as fast as it can so that you don't keep overstimulating the muscle with information. The acetylcholine gets broken up, you recycle part of it, and you burn up the rest of it through a cellular respiration process. Okay? All right. That acetylcholine is commonly used. This is the system that we're dealing with. All right, now what I want to do is just highlight a little bit about toxins. There are lots of organisms out there that make toxins, and a very common thing to do for organisms is to make toxins that target some kind of signal molecule or signal molecule system that either involves communication between a neuron and a muscle or between two neurons. Okay? Now, why would you make toxins to target these systems? Well, it's a pretty quick way to disable an organism. If I'm a snake and I'm trying to eat this mouse, I don't have any arms. It's not like I can hold it down or strangle it. If I don't disable this mouse's muscles, its claws and teeth are just going to shred me to pieces. So I make a toxin. Many snakes make a neuromuscular junction toxin. And that neuromuscular junction toxin immediately targets the neuromuscular junction of the mouse and disables the ability of the neurons to get the muscles to contract. So by disabling that system, I've disabled the ability of the mouse to bite me or claw me. Okay? Many animals make these toxins. Plants make tremendous numbers of toxins. Many of these are neuromuscular junction inhibitors. Your typical plant, you go outside, look at an oak tree, figure about 350 unique toxins. And that's why there are only a few plants that we actually consume as humans. The vast majority of plants were taught from a very early age to leave them alone. Don't eat them, spit them out, right? Many plants target neuromuscular junction. Fungi target neuromuscular junction. Bacteria target neuromuscular junctions. So all these organisms have come up with different ways to target neuromuscular junction. So what I want to do is just talk about a couple of these in a very broad sense so you get a feel for them. And my guess is those of you moving on in medical fields, this becomes a larger discussion at some point in the future. Might help to have a little bit of information going into that. Okay? All right, so let's do some of these neuromuscular junction toxins. So the first one here is tetanus toxin. This is made by a bacteria specifically Clostridium tetani. This is a bacteria that prefers anaerobic conditions, so it's common in canned foods when you haven't gotten rid of these bacteria properly, and they end up in the can, and eventually the oxygen gets used up in the can, and then they go to town making lots of this toxin, or think like a deep puncture wound where something's been punctured in here, and under the appropriate conditions, this portion of the wound can become anaerobic because it's difficult for the oxygen to come in there. Okay, that's probably the more common one here. Is it's been driven down into the skin to a point where it's difficult for the oxygen to get there, and now this tetanus toxin is produced. All right, the tetanus toxin here inhibits the central nervous system synapses. Okay, and specifically the ones that inhibit something. So we're talking about synapses that release glycine or GABA. We talked about these neurotransmitters before. That these neurotransmitters are released from these central nervous system synapses. The tetanus toxin is targeting these synapses and preventing the release of these inhibitory neurotransmitters. This is the one I was thinking when I was talking about tetanus, because it's also a Clostridium. The botulism toxin is made by another anaerobic bacteria. It's botulinum? Botulinum, yeah, botulinum. This is the one that ends up in canned foods a lot. The story is, I think, a gram of botulism toxin is enough to kill everybody on the planet. It's a very toxic molecule. This particular one inhibits neurotransmitter release. Just like tetanus toxin prevents release of these. In this case, the release is accomplished by preventing vesicle docking. So the neurotransmitter is in that synaptic terminal. I need to communicate with somebody down here. I prevent the vesicle from binding to the membrane. I prevent the release of the neurotransmitter. So the Clostridium has shut down the interaction of this neuron with the effector cell by preventing the release of the neurotransmitter. Okay? All right, so this first model is prevent the release of the neurotransmitter. If you block neurotransmitter release, you block the signal from ever getting to the effectors. Okay. The second model I've got is more of a receptor inhibitor model, where we're going to somehow mess up the receptors. Okay? So let's look at a couple of different types of receptors. When people first worked out the receptors that were present on the effector cells, they were able to work it out by using toxins. So if a particular receptor was affected by this toxin, they named the receptor based on the toxin. So there are a group of receptors on effector cells called muscarinic receptors, and that's because these receptors are susceptible to a fungal toxin called muscarine. There's also a group of receptors called nicotinic receptors. These are susceptible to the toxin nicotine. There are also cannabinoid receptors, which are susceptible to the cannabinoid toxin from marijuana. Right? We could go on and on and on listing out all these different types of receptors. But the muscarinic receptors, these are fairly common at parasympathetic synapses. So this is where the parasympathetic nervous system is communicating with an effector cell. Okay, so think heart, intestines, right, central nervous system, right? Communication is the parasympathetic. And then you should be able to remember what the parasympathetic nervous system does. I'm not going to give it away yet, give you time to think about it. And then the nicotinic receptors are found at neuromuscular junctions. This would be nervous system combining with skeletal muscle, at that conscious muscle. Okay? All right. Now, let's take a look at a few different toxins. Here's curare. That's made by a frog. I think it's a poison dart frog. I'm not sure if the frog sequesters the toxin for a plant. Right? In any event, this is the plant that makes it. Strychnos toxifera. Fera is bearing,